We often treat the definition of a plant as a static historical truth, an obvious boundary that has always existed in nature. However, the map of the plant kingdom has shifted significantly since the mid-20th century. Under the five-kingdom classification proposed by Whitaker in 1969, the definition of plantae included several groups that would be unrecognizable to modern biologists. Early taxonomists relied on a narrow set of observations, grouping fungi, monera, and protista within the plant kingdom, simply because they all possessed a cell wall. As our understanding of cellular biology deepened, these groups were definitively ejected. Fungi and cell wall-bearing microbes were moved into their own distinct kingdoms, leaving behind a more refined definition of what a plant actually is. This update even affected organisms long considered to be plants. Cyanobacteria, or blue-green algae, were stripped of their algae status entirely when they were identified as bacteria rather than true plants. Biological classification is a dynamic, self-correcting algorithm that shifts as our data resolution improves. The modern plant kingdom now consists of 5 core groups: algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. With the kingdom's boundary set, the challenge for taxonomists shifted to mapping the internal relationships between these immensely diverse flowering and non-flowering groups. The first scientists to attempt this mapping were constrained by the limits of the naked eye, categorizing nature based only on the data they could physically see. To understand the architecture of modern botany, we must trace how biology transitioned from these superficial observations to deep structural data analysis. The earliest phase of this effort produced what are known as artificial systems of classification. These systems were strictly limited, relying purely on gross, superficial morphological characters to distinguish between species. This illustration of unfolding leaves highlights data points early taxonomists logged: general habit, color, and specific leaf shape. Linnaeus's system was heavily reliant on these vegetative characters and the basic structure of the andrecium, or male reproductive parts. These inputs created a low-resolution map where organisms were grouped together based on surface-level similarities. By relying on a fraction of available biological data, artificial systems frequently split highly related species whose exterior traits happened to differ. A primary flaw in the artificial system was that it gave equal statistical weight to vegetative characteristics and sexual characteristics. This weighting is unreliable because vegetative characteristics like leaf size or color are highly unstable. They are easily altered by the environment. In contrast, sexual characteristics remain structurally stable, providing a consistent signal regardless of environmental noise. A robust biological algorithm requires immutable internal data rather than variables that change with the weather. This led to the development of natural classification systems. These systems prioritize deep natural affinities among organisms over coincidental external matching. This required a significant upgrade in data resolution as taxonomists began analyzing internal ultrastructure, anatomy, embryology, and phytochemistry. George Bentham and Joseph Dalton Hooker successfully deployed this high-resolution natural classification specifically for flowering plants. By examining the internal anatomy of an organism, taxonomy moved from a sorting exercise into a rigorous structural science. Today, the universally accepted standard is the phylogenetic classification system. The logic here is based entirely on mapping evolutionary relationships rather than just physical similarities. Under this system, organisms placed within the same taxa must share a verified common ancestor. Modern taxonomy acts as a mathematical reverse engineering of the deep history of life on Earth. The primary obstacle to phylogenetic mapping is the fragmentation of the fossil record, which leaves many ancestral links undocumented. Without fossil evidence, taxonomists must find other ways to prove that two plants share a common origin. Numerical taxonomy uses computers to assign codes to hundreds of traits. This gives every character equal importance, bypassing missing data and removing human bias. When physical evidence from the past is unavailable, overwhelming computational data can calculate evolutionary proximity through statistical probability. To resolve difficult classification errors, taxonomists utilize cytotaxonomy. This method relies on internal cellular data, specifically the number, structure, and behavior of chromosomes. The most granular level of analysis today is chemotaxonomy. When physical structures look identical, chemotaxonomy analyzes the literal molecular signatures and chemical constituents of the plant to resolve the classification. Botany has evolved from a debate over the shape of a leaf into a multidisciplinary data matrix. By combining computational, chromosomal, and chemical analysis, we are finally architecting the true evolutionary map of the